Hello, everyone. My name is Jaswinder Singh. I am an associate professor at the McGill University in the Department of Plant Science. First of all, I would like to thank the organizing committee for inviting me this plenary lecture today in this very prestigious conference, fourth International Scientific Conference, which is happening in Ukraine on the latest achievements of biotechnology. So I especially thank Dr. Nadia Adam-Chakchala, who approached me for this particular seminar. I get to know her last year when she visited us at the McGill University and delivered an excellent seminar there. So I found many commonalities with your institutes and our institutes where we are working to improve our croplands. So today I will deliver a seminar on a transposon-assisted biotechnology in the small grain cereals. So there are many approaches which we are using in my laboratory at the McGill University to improve the croplands. Before I do so, I also want to congratulate the organizers to do this conference, even when we are going through very unprecedented times where people are pretty much nervous about the pandemic. So this is really a courageous decision by the organizing committee, and so that we can continue to provide information to our fellow researchers, students, and colleagues about the latest developments in the biotechnology. So I hope everyone is safe and healthy at your place. Although I cannot able to meet you today in person, however, I will be talking about some of the latest approaches or different techniques which we are using to improve our plants. So before I do so, there are few words about the McGill Universities. McGill University, many of you might know McGill University. This is a highly prestigious university in Canada, which is ranked number one among the medical doctoral universities. And recently, it is ranked 31st based on the QS World University ranking at the global scale. So we have 11 faculties and 11 professional schools, and I am working in one of those faculties, which is known as Faculty of Agriculture and Environmental Sciences. So we have more than 40 students here. We are teaching, who are actually, studying here all over the world, and we have a lot of international students in our university. So, but we in McGill University, we have two campuses. One campus which you can see behind my face, that's a downtown campus, which you can see there's a very nice fall behind it. But we have another campus which is where our Faculty of Agriculture and Environmental Science located. And on the slide which you see, this is the St. Andy Bellevue, where this campus is located. You can see a beautiful campus, and I invite everyone to visit this campus, especially in the summer, when you can see a very nice scenic view, which you can observe from my slide. So if you want to see the aerial view of our campus, it's also known as McDonald campus of McGill University. This is an agricultural campus, and you can see in the summer, this is the aerial view of our campus. So I'm sure you may be excited to see some of these nice pictures and probably visit us sometime later and talk to us about our programs. So, okay, let's try to discuss about some scientific research. And as I said, I will be talking about the crop improvement through biotechnological approaches. And now onward, I will talk about targeting genes for cereal improvement. And you may think, why the cereal improvements? There are so many other crops on which we work one way or another. So why we work in the cereals in my lab? The main thing is because, as you know, cereals are critically important, and they are important food sources. The vast majority of calories which we consume, they are coming from actually cereals. So they are coming directly from plants or indirectly from plants via animals, because animal also eat plants. So basically most of the calories are coming through the cereals. And whole grains, now which is becoming very, very important, they are very good sources of carbohydrates, dietary fiber, and fatty acids. So over 70% of the world's harvested area is planted to the cereal. So you can see the cereals are key to the global food security. So my lab is basically working on that. And if you see this chart, we produce approximately 2.6 billion tons of cereals actually, and which feed the whole world. And but if we, as the population is growing, and it is projected that in during approximately in 2050 we have to double the cereal production to feed the whole world. So basically we have a big challenge on us, how we can do this, although our areas being reduced. So basically this is a big challenge. So we, all our scientists, they are working day and night, thinking about different strategies, how we can double this production in next few years, next few decades, so that we can feed the world. And so you can see the cereals are so much important for the food security. And so basically there are many approaches which we can use to improve it. But before we use any approach, so basically we need to know what's happening. So means how we can do, which approach we can use. But there are so many challenges. There are significant yield decline. We are projecting, which is approximately 20 to 30% due to the climate changes, through abiotic stresses such as heat, drought, and salinity. And there are many biotic stresses as well, which we need to actually develop resistance to. So when we check different strategies, and we can see the genomics and biotechnology could be very helpful for the development of next generation of these high yielding crops, and to make safe crops and nutritionally rich crops, and which could be climate resilient cereals. So for this, when we are talking about genomics and biotechnology, so definitely we have to identify first genes, good genes, which we can actually introduce in our crops, so that we can improve these crops further, which become climate resilient, and which become more resistant to biotic and abiotic stresses, and and give us higher yield, and so that we can double the yield of cereals. So there are many different strategies. As I said, there are new functional genomics tools. Functional genomics mean you can understand the function of the genes we can use to identify good genes, so that we can make our breeding effort reliable and efficient. So in a nutshell, I can say continuous research efforts in crops genetics, genomic physiology, breeding, agronomy are required to meet the future demands. So we have to use whatever the tools available, and and biotechnology and genomics is one of the tools, not all of the tools, which we can also integrate while we are improving the crop plants. And I believe this is also one of the theme of your conference. So new biotechnological approaches have a great potential to do this and to help in the food security issues. So now I will talk about what kind of tools we are using currently to identify function of the genes. So you can see there are two pictures on your slide. One picture looks like apples, and other looks like the spike of a cereal. However, in one side, this is not the apple. This is a little bit bigger format of the corn seeds, where you can see different sectors. And many of you might have already know what those sectors are. Actually, these are based on some of the jumping genes, which actually identified by Barbara McClintock long time ago, and she got a Nobel Prize. So what she found in her amazing discovery, there are two type of elements, which are dissociation or activator. You know, at one time there are only one element. In evolution they become separated. So basically, with the help of activator, dissociation element actually move from one place to another place, and it entered into genes. When it entered into a gene, then it actually stop its function. So it interrupts its function. When it interrupts its function, so you can go back and see in which gene it is interrupted, and you can find, okay, this is the gene which actually does this thing. Let me give you a very simple example. For example, if this is your DNA, and for example, if there's a gene which make this blue color, and if somehow you interrupt this particular gene with the transposon, the DS or dissociation element, its color becomes yellow. So from this, one can just infer that this particular gene is actually involved in the color development. This is just a simple textbook type example, so that you can understand how we can use these elements to understand the functions. Once we know the function of the genes, then we can use those good genes for our crop improvement. So in my lab, we are working on these elements, and we are working on in barley. We were the first originally when I started my postdoc at UC Berkeley. At that time, we have started working on the transposon system in barley. Why barley? Because barley is the fourth major cereals worldwide, and it is used in animal food. And you can also say the barley is a beer crop. So people love barley because of beer. So barley also used for human consumption due to its health benefits. And we know now that using barley in your dinner table, it can lower the blood cholesterol as well. So these are the general important things about the barley. But barley also actually a great crop to do genetics, because barley is diploid. It is a very good model for a wheat crop, and wheat is one of the key crop for the food security. Whatever we understand by doing a research on barley, we can clearly translate that research to wheat, because all the 7 chromosomes which are present in barley, they are these chromosomes are pretty much homologous to the wheat chromosomes. They are pretty much similar to the chromosomes of wheat. So, and also in barley, we have multiple genetic resources available, for the whole genome sequences available. If we know the sequence and then we interrupt with these kind of elements, transposable elements, you can understand their function. And once you understand their function, you can use in crop improvement. So to do this, we start from tissue culture and transformation. So what we do, we use immature embryos from barley, and we develop a callus, and we develop some specific type of callus which is known as a green callus. And that green callus, we introduce those transposable elements through the bombardment gun, which you can see in the picture. By this is also known as a ballistic method. When we are introduce those elements into the callus, then we select them based on the different reporter genes, and in the end we develop plants where we already know these transposable elements are actually inserted. And then we see whether these transposable elements are actually jumping around or not. So they randomly jump in the genome. Once they jump into the genome, then you see the phenotype. And when you see the phenotype, then you can go back and see which gene is interrupted, and then you can connect that gene with that phenotype, and then that's how you can understand the function of the gene. So how this two component transposon system works, and if you see on the slide, you can see this is, for example, a cell, and this white color is all cytoplasm, for example. And in that cytoplasm, there's a nucleus, which is a pink color, and all different bars are there, 7 different chromosomes of barley. For example, you introduce a DS element by transformation, which I just shown to you with the bombardment gun. You can also introduce an activator element, with the transformation. When they come together, what happens in the presence of this activator, when it is making an enzyme which is known as transposase, the other element, they jump from one chromosome to another place. Sometimes they do not integrate, but when they integrate, sometimes they integrate very closely where they were originally located, and sometimes they land actually in a different chromosome. In this way, you can develop a single copy transposon ancestors line, because in the next generation, those single copy which are away in the different chromosome, they are segregated away from the other material. So in this way, you have a mutant in the single gene, developed through this transposon system. You can also develop lines separately. For example, you have one transformed line with the DS element, for example, line number 1 here, and line number 2 you can also develop separately, which has an AC element. Once you have that element, then you can do a breeding, you can do hybridize them, you can cross them, and then you can develop a population. And the population, when they become together, as I have just shown to you in a previous slide, in the presence of activated DS elements move, and then it can go to many different places. This is just an example. When you check them, their RFLPs in a southern blot, you can see all different bands are actually due to the presence of element in a different gene. So which gives you great evidence that these are some of the single copy, as you can see. And based on that, we have developed now more than 300 unique lines just to understand the function of genes. And some of the examples I will give you today, where we have already discovered some of the functions because of these lines. So we also, when we interrupted these gene sequences, then we know the sequencing data is available. You can go back and check in which genes they are actually landed, and then you can see what's the structure of those genes, where those introns are, where those exons are, because we will be more interested if they are interrupted in the exons, so that the expression of those genes will be affected. Then it will be a little bit more easier for us to understand the function of genes. These are, as I said, we have developed more than 300 lines. This is a subset of lines where we have actually, analyzed these mutant lines. So let me give you one example, which we have published previously, and we have actually developing it further, and one of our manuscript is actually under review. Basically, so we have identified a gene which is known as VAC1 gene. We also call as wall associated kinase 1 gene, which we have found that actually that particular gene is involved in the root length. We have some evidence that this particular gene probably may be involved during the salt stress as well. So that basically, this is just an example from this repository. We found some of the interesting key genes where, and we are understanding their function. So that basically, so we also have another line, transposon insertion line, where actually we are understanding the role of microRNAs. For example, there are different transcription factors. For example, SPL genes we also call as squamosal promoter binding like proteins, which actually are interact with the different microRNAs. So we have one of the transposon line where MIR172, one microRNA gene, is actually interrupted by this DS transposon. And based by analyzing those, we found that this particular line is actually gene or microRNA is involved in the spike development. So basically, I mentioned to you a gene which is involved in salt stress or the root length, and I am also mentioning that there are genes which are involved in the spike development. So means there's a different range of the phenotypes which you can understand by this particular mechanism. So another advantage of this type of transposon assisted biotechnology is that when you interrupt something with the transposon and then you move away that transposon from that particular gene, it's again cause a very specific edit in that particular gene. There will be 3 to 4 base pair missing in that particular gene where this transposon was originally located. If you cross that transposon with the activator line, transposon will move away and segregated away as well. And what you have left in your hand, that a plant where the gene in which the transposon was landed now is not anymore there. However, there are some small mutations, and I call it as a transposon-based gene editing when we reactivate those transposon insertion lines. So basically, so this is, as you know, the CRISPR or gene editing is pretty popular in these days. So this transposon-based gene editing, although it is a little bit more random, it's not, we can also use transposon system to develop those kind of edits in plants and then see its actually effect on the phenotype. And I'm not showing a slide here, but we have seen some of those edits which have an effect on the grain germination, and the small effect on that gene affect the germination. Another use of this transposon system is that, as I said, that when the transposon is inserted in one gene, it interrupted its function. Sometimes because of the redundancy, you cannot find the phenotype. Another way you can approach is that you can map them on the chromosomes. We already have mapping resources. Sometimes, as you can see, there are different chromosomes, for example, 4H or some 3H, they are Barley chromosomes. These all small, pale green color, boxes actually indicating that where those transposon insertions are mapped. Some are close to some very important, genetic loci which we know they are involved in either in disease resistance or abiotic stress resistance. So which when you reactivate them again, then as you have seen in one of my previous slides, that sometimes it lands very close to that. And it happens more than 70% cases, it landed very close to on the linked position. If your transposon insertion is mapping very close to one of those important gene area, chances are very high when in the next generation, when you reactivate these transposon insertion lines, they will we have a great chance that they can capture the gene which is in that region, and we can then connect them with biotic stresses or abiotic stresses, and that's how we can use them as a map based saturation and mutagenesis. And I'll give you some examples of that in the later part of my presentation. So basically, as I said, what we did is, for example, if you see on the slide, there are two insertions of transposons which were mapped on a chromosome 4H, which you can see here in the barley, and one of the region, which is a telomeric end of the chromosome 4H, there is a QTL, this is a quantitative trait loci, which is known that this particular QTL is involved in a malting quality. As I said, barley is a beer crop. We want to identify what are those beer genes are. So, and this particular QTL was very hard. And originally it is identified by, Pat Hayes' lab in 1997, where they located it on chromosome 4H. And then in another lab in Washington State University, Professor Andy Kleinhoff's lab, they actually fine mapped it in 2004. And what we know about this QTL is that more than 29 to 38% variation is there for malt beta glucan and malt extract. So these are some of the key components which influence malting quality of the, means beer quality of the barley. So interestingly, the lines which we have developed through transposon insertions, which I said we have more than 300, and two of the lines, especially one of the line, TNP29, which you can see here, actually mapped in that particular or close to that QTL. And that gives us a very big hope that if we reactivate this, and probably we can saturate this particular region and find the genes just like this. For example, this is a malting quality QTL2 and TNP29 is mapped there. And there's another one which is a little bit further away, but it can also be helpful. But our main idea is that when we reactivate them, these transposons insertions, our aim was just to saturate that region with our transposon and then see in those lines what the phenotype is. If the phenotype we found which is affecting malting quality, then we have something special in our hand so that we can use those genes and to understand their function later, how they are actually involved in the malting quality. So we use two different strategies to explore this particular QTL2, which is involved in malting quality. So one was actually transposon activation, which I just explained to you. And then the aim, again, I want to show you in a different way that this is TNP29, that's a QTL region. What's happening that when we reactivate them, so this transposon will move from one place to another place in many places, but we will be more interested in all those transposon insertion lines which are actually mapped or landed in that QTL region. So that we can, further analyze them and, understand the function of this QTL2 involved. And we were we have done very successfully. So we crossed that particular TNP29, which is a transposon insertion line, crossed with an activator line, which is we call it 25B. We created a population. We identified some of the new lines and then checked their mapping location and find out which one of those are actually mapping in that particular region. while we doing that, we have identified many new mutations, and we also, noted that uh, basically approximately in 92%, 9 to 11% reactivation frequency, which is I believe good enough to find the lines in which you are interested. In addition to that, we also used some of the other approach, because one of our transposes is actually fused with GFP. So in order to find out which line will be making that enzyme, transposon enzyme, we used in vitro reactivation approach. So instead of crossing them, what we did is we are giving them transient shock, of activator transposase enzyme, which is fused with GFP, and select those where the GFP is expressing, which you can see after 3 days of, and after 70 days of inoculation. And then we select those, and by this we have increased its efficiency 3 times. For example, now you, instead of 9 to 11%, it's basically approximately 30%, frequency of transposon, which is pretty good news, which may be very helpful to identify some of genes. So these are some of the lines which we identified through this, and most of them were actually, many of them were mapped in QTL2 region. So while we were doing while we are analyzing them, so somehow we cannot able to identify the line, or the because we need to identify more lines in order to identify the real gene. In the meantime, so we thought we can also use another biotechnological approach, which could be based on the synteny based. So what we did, we know the region of the barley which is actually involved in malting quality. We took that region, and we, at that time barley genome was not sequenced, we matched it with the rice genome and find some of the candidate genes, just like this. So we know the barley 4H, this region is actually, you can see in the boxes, it matches to the genic region, which is boxed in the red chromosome of rice 3 chromosome, in the rice chromosome 3. And based on that, we identify more than 100 genes. Now, how you can pinpoint which gene is actually involved? So then we narrowed down 22 putative candidates which are involved during germination or malting. So only those genes we selected. And one of the gene we hypothesized that it could affect the malting quality. And this is another picture. We have 22 genes, as I said, identified, and we named them MSB1 to MSB22, and MSB12 gene, which we identify based on the gene structure, it has given us idea that it could be involved in malting quality. The reason is because when we analyze this gene, we found there are some binding motif, which have a sugar binding domain, which can bind to the beta glucan or those kind of carbohydrates. Because originally, if you go back to my previous slide, I mentioned this particular QTL is involved in somehow the amount of beta glucan, which is actually influenced from that particular region. So it gives us pretty much big hope that it could be this gene may be involved. Then we start exploring this gene further, and we found this MSB12 actually belongs to a thaumatin-like protein, which is actually a part of the pathogen-related gene family, which has many pathogen related gene families. There are 17 of them, and this particular gene is actually a part of the PR5 gene family. And so we also know these gene families are involved. And there are many genes which are part of the PR5 gene proteins, and they are involved in many things. And interestingly, this thaumatin-like proteins, why we call them thaumatin-like proteins, because originally they were isolated from the Tomatococcus danieli fruits. They are sweet in taste, and so and these are 21 to 26 kd proteins, and they, interestingly, they have lot of disulfide bonds. They are rich in cysteines, which helps to develop some disulfide bonds. As I said, the thaumatin-like proteins or PR5 proteins, there are many functions we are involved in: fungal pathogen, drought, salinity, seed germination. And so in our analysis, we have added a new function to this MSB12. And once we explored it, we found out that this MSB12 is actually TLP8, thaumatin-like protein 8, which actually mapped on chromosome 4H. That's where exactly our QTL2 is. And it has binding motif, which can bind to the carbohydrates. And so it becomes a very interesting candidate for us. So then now we have a sequence in our hand, and we started taking 3 varieties which are feed varieties, 3 varieties which are malt varieties. Difference between malt and feed varieties of barley: in the malt varieties, we always use those which have a little bit low beta glucan content, because high beta glucan content affect the filtration of beer, because it makes the wort very viscous. So basically, we took 3 varieties of malting, 3 varieties of the feed, and we checked its expression. When we checked its expression, as you can see from this graph, all the high bars, it expresses higher in the malting varieties, but it expresses lower carboacylation and strepto in the feed varieties. So, which indicate to us that expression of this particular gene is actually involved in the, it has some relationship with beta glucan. When its expression is higher, so beta glucan content is actually lower. When its expression is lower, its beta glucan content is higher. And beta glucan content, I want to mention here, is that it's not only have a role in the malting quality of the beer, because here it has a negative role, that it makes the wort viscous. However, if you check as a health point of view, the higher beta glucan diet, which is a part of the dietary fiber machinery, and it is very good for the health things. So means with that particular gene, by changing its expression, you can do 2 things. One is you can develop varieties which are very important for malting quality, and second, you can develop variety where you can express it more, where we presume that beta glucan content will be high, and which will be for the health point of view, those varieties will be excellent. So when we checked in all these different varieties, beta glucan content, interestingly, we saw within 48 hours, 16 to 48 hours, in the malting varieties, its expression was drastically or significantly reduced. However, not in the feed quality. So basically, which indicates to us that it has a lot of relationship with the beta glucan, because if we use those feed variety in those malting process, our wort will be highly viscous, because this particular gene is not expressing well. It cannot bind to the beta glucan, and based on this information, we are thinking that this particular gene is a glucanase, probably a glucanase, which is actually acting on beta glucan, and it chopped it up. How it does, we don't know yet. So, but if its expression is low, the beta glucan remains in the wort, and it affects viscosity. So, but we can still use for the feed varieties. So means this is just an example how we can use different biotechnological approaches to understand the function of the gene. So because we want to study it further, because this is very interesting. It has a lot of cysteines, and when there are cysteines in a protein, they can come together, and they can, with the help of the disulfide, they can create a lot of disulfide bonds. If the more disulfide bonds are there, more cysteines are there, sometimes those proteins are more stable, because the glucanases which we generally add during malting, many of them, they are not very heat stable. So as it has a lot of cysteines, and we predicted that probably these glucanases may be more stable. But before we reach to that point, we really want to see how this particular protein is actually acting on beta glucan. So what we did is we cloned that particular gene, and we expressed an E. coli. We got the protein, recombinant protein, and then we did some experiments that where we put the beta glucan and protein together, but in a different environment. In some cases, we are giving them, oxidizing agents. In some cases, we are giving a reducing agent. And we found out, and that's what our hypothesis was, because it has a lot of disulfide bond, cysteines are there, and probably with when we were giving them a reducing agent, so means its conformation may change and it may not be active. And we found exactly the same thing, that when there is a reducing agent, for example, when you put the DTT or other reducing agent, so means it's binding with beta glucan drastically or significantly reduced. So means basically, which indicates to us that this particular protein, when it is present in the system, it also it is redox regulated. It needs a appropriate appropriate environment to interact with the beta glucan. So you can understand, we started from a chromosome, a region, and by studying using different biotechnological approaches, now we are digging deep and identifying which gene is actually involved in the malting quality and how it is involved in the malting quality, So basically, this is a beauty of the biotechnological approaches. Now, this particular genes, we are recommending, or we are using and developing some markers on it, so that you can use, based on this gene, we can develop some varieties which will have a better malting quality in barley. So this, based on this work, which is 2 years ago published, we made a hypothesis that if there is a high expression of TLP8, this particular gene, there will be more binding with beta glucan, and there will be less beta glucan in water because of the glucanase activity probably, and it will improve the lautering and quality. However, if in the feed varieties, its expression is lower, it has a less binding, and there will be more beta glucan in the wort, which will have a serious consequences. That's why those varieties which have high beta glucan, which we generally do not use in the malting process. So conclusion for this part of the study, that as you can see, we have successfully developed, transposon-based mutants in barley, and using that mutants, we have identified the function of many different genes. I have given you an example of the microRNA 172 with the spike development. I have given you an example of also a wall-associated kinase gene, which is actually involved in the root length, and also given you, based on the saturation mutagenesis approach, how we can even understand the function of a QTL and identify the specific genes or major key genes present in that QTL with the help of transposon, as well as by the synteny-based biotechnological approaches. So finally, what we found, that the QTL2 region, which is the region on the chromosome 4H, harbors a key gene which is known as TLP8, which is a part of the thrombin-like protein PR5 gene family, and which actually interacts or bind with beta glucan through carbohydrate binding domain, and its bindings actually is influenced by the redox level during the germination. So means these are some of the key findings from our transposon work in the barley, and fortunately we have successfully published this work in PNAS in 2017, which is one of the high profile journal. And this studies also got a lot of media attention around the world, that this is sort of a breakthrough in the malting industry. So now I will shift some gears. Not only this crop, this cereal we work to decode genes. So beta glucan, as I said, it has a second angle. If you have a more beta glucan, it is a high dietary fiber, which will be good for health. So as you know, in your breakfast, many people use oatmeal, right? There are many breakfast meals are oat based, and oat is becoming very now prominent cereal, especially because of these health concerns. So then we thought, okay, we are successful in barley. Is it possible that we can do the similar things in another genome and another cereal and become successful? And we selected oat for this because of, as I just said, because of this dietary fiber stuff. So there are a few more slides which I will show you for a few more minutes, where I will show you the same transposon assisted biotechnology, how it is helpful. And we have not published this yet. This is a very, very new work. So why oat? As I just said, that is an important, for food and a feed crop around the world. And it is considered as a health food because it can specifically lower the LDL level, right? So, and another thing in our mind was that there are limited genetic resources are available in oat. So means we are interested, because we have those skills, why we cannot help the oat community further to develop such resources. And we started developing a transposon assisted biotechnological approaches and functional genomics tools in oat. So idea here is that, based on that, we are interested to identify novel genes which are actually associated with dietary fiber, oil content, disease resistance, and agronomical traits. Because just like, so that jumping from one place to another place to, trap those genes, to target those genes. In a nutshell, I can say we are developing a gene tagging resource in barley. So it is definitely based on our, as you have seen, we are very successful in barley. In many other places, some other people also have done work in barley. Researchers already have done in Arabidopsis. But there is one different approach we are using, which other people also have used. So means if you see oat and barley is diploid, right? But oat is hexaploid. If it means in oat, there will be a lot more redundancy. Even if we interrupt one gene, there will be another copy from another genome in oat, because it's a hexaploid. So by knocking out one gene, it will be very difficult for us to find a phenotype. Although in barley, the phenotype will be a little bit more easier. So then there is another transposon-based approach that maybe in the end of those transposon construct, we can add a strong promoter, which people have already done in Arabidopsis, maize, and rice. So we use that approach so that when it will jump from one place to another place, where it will be landed, the strong promoter close to that where it is landed, if there is any gene, it will overexpress that. That's known as activation tagging. Instead of knocking out the gene, we are activating the gene. So and in this way, which will be useful if this higher expression, with that high expression, we will know if it has an effect on on the phenotype, and then we can see which gene is actually affected, and in this way we can understand the function. So this is the reason we used a different type of approach, although we are also developing a knockout approach as well. Excuse me. So to develop some functional genomic resources. So before we do in a new crop, we have to actually check its efficacy. So we tested its efficacy with the transient assay. For example, if you see, this is a transposon construct. This is a, after transposon, there is a Gus gene, right? And here, and this is a sort of, this is a promoter as well. So basically, if this particular part is moved, and with this promoter, Gus will express. This is a transient expression analysis. So for example, if you, it's jumped out, and the Gus will express. If the Gus is expressing just like which we have shown, it means our system is working. So mean in this particular genome, this system could work. So we were successful in that. So then we have developed some activation tagging construct. Actually, those construct we also got from Dr. Mike Iliff from CSIRO Plant Industry, Canberra, Australia. So we use those which they have published in barley. We use the same construct in oat. So you can see, for example, if this is the DS element, and before DS, there are strong promoters, UBI promoter, which is transcription in this direction, and UBA promoter on that side as well. Transcription is going that direction. So basically, when they will land on a genome, which I'll show you within a minute, and the adjacent gene will overexpress, just like this. For example, it is landed, for example, adjacent genes are, expressing. So with that, we also develop this system in oat as well. So we use the similar way as we have done in barley. The only difference is that in barley, we have developed the callus from immature embryos, but here we have used the callus developed the callus from the dry mature seeds, and it worked beautifully. And using with the same bombardment gun, we introduced those constructs into oat germplasm. So oat genotype, the oat genotype which you used is named as PARC, which is in the literature is known as, which is very good for transformation in oat, because not every genotype can be transformed in different cereals. So when we did it, as you know, in a construct, its Gus is very close to that, and so we saw Gus expression, and we were very successful. So basically, it indicates to us that our transformation system is working, and these are the stable lines. So we also tested, because one of the reporter gene, one of the antibiotic selection gene, we have a herbicide resistance gene actually in a construct, which is also known as bar gene. When the gene is expressing, if we painted with the herbicide, you can see all resistant, but in the control, they are all susceptible, which also indicate again that our transformation is successful. Third thing we did is, because we also use other different constructs, for example, which have GFP. And, when we introduced those and we saw there is a GFP expression in the transgenic calli, so it also indicates that, okay, our transformation system is pretty much working in PARC variety. And now we are ready to introduce our transposon constructs there, for a stable transposon system, the same way which we have developed in barley. So we developed it, and we have developed many transposable transposition events. These are some of the examples, and we found somewhat a little bit higher or approximately similar, but that was 9 to 11%, here 12 to 14% transposition frequency. So it's working beautifully. We have not published it yet. So these are some of the new results I thought I should share with my colleagues and my researchers. So and fellow researchers and students. So our conclusions are that yes, we can successfully transform oat species with the ACDS constructs. So, and this transposon system is fully functional as a transient level as well as at a stable level. So I'm not showing the data here, but when we checked the flanking sequences, the where the gene is at, the transposon is landed in the oat genome, we confirmed that we tagged the oat genes. So we will integrate this system with the other available resources, and even with the gene editing, and to develop a fully fledged, fully workable functional genomics resource in oat, and which will help to understand or identify genes which I just mentioned, which are involved in dietary fiber, in disease resistance, in abiotic stresses, biotic stresses, oil content. And it seems like that this resources in hand will be very useful. So this is the actually first ever effort to introduce heterologous transposon system into its genome, oat genome for functional genomics. We are very hopeful that it will be very useful for many people who are working on serial biotechnology. So in the end, thank you very much for listening me for this talk. I'm not present in person, but so that I should answer your questions, but feel free to send me email if you have question, and I will try to answer them. So before I wrap up my presentation, I want to acknowledge many people, many students and postdocs who worked with me in my laboratory at McGill University, who have contributed one way or another in many different projects which are based on transposons. And so their pictures are here. Many of them, I don't want to take much of your time to express who what did what, but all of them one way or another they have contributed. So in addition to that, no research can be successful until you have a funding. So I want to acknowledge all the funding bodies which I'm mentioning here, so who has given us funds to do this interesting work. And recently we got a federal government prestigious funding for a CREATE program, which you can go www.jeffsess.com and see what we are doing. It is a $1.65 million grant from federal government, where we are actually now developing other biotechnological approaches, not only plants, but also in animals based on the gene editing. So with these words, thank you very much for giving me an opportunity so that I can talk to you, about the research which we are doing at the McGill University. Again, my contact information is on this slide. You can send me an email if you have a question. You can also explore our different websites to understand, to explore what we do at the McGill University. With these words, thank you very much. Stay safe, and feel free to contact me. Thank you. Bye. Bye for now.